Good afternoon, everyone. It's my pleasure to welcome you all today to today's Nanomedicine Symposium. My name is Nazila Kamali, and it is my pleasure to kick off this LCN Symposium today. We are delighted to have such a fantastic lineup of speakers this afternoon, and we thank each of them for agreeing to talk at this event. And of course, many thanks to all our audience joining us online from all around the world. We hope that given the range of the breadth of the talks today, that there is indeed something of interest for everyone. As you already know, the last year or so has seen extraordinary strides being made in the area of the application of nanotechnology to healthcare, where we saw the Pfizer, BioNTech, and the Moderna COVID-19 vaccines, which are based on mRNA embedded within lipidic nanoparticles, be rolled out and approved for human use in unprecedented time frames of less than a year. And we can all agree that these nanoparticles have been pivotal in saving hundreds of thousands of lives across the globe this year alone. I'm sure that we can appreciate the huge nanotechnological feats involved here relating to scale up of the nanoparticles and this unprecedented scale of being able to store and stabilize these particles and meeting the challenges in particular of storage. It's been amazing to see how well this has all been handled and accelerated with an extraordinary level of coordination and collaboration across disciplines. As nanomedicine researchers, we can now hopefully really look to the future with a much more positive perspective. And therefore, with this timely symposium today, we would really like to highlight some excellent nanotherapeutics, nanodiagnostics, and nanotheranostics research being conducted across the London Centre for Nanotechnology affiliated universities. In addition to our own fantastic keynote speakers who will be joining us from Silicon Valley this afternoon to give us a more translational industry perspective. So before moving on, I would also like to thank my colleagues Yuval Elani, who will be co-chairing the sessions with me today, Hafisa Bibi for her help with organizing this meeting, and a huge thanks to Sandrine Hoytz, who is the co-director of the ASCN at Imperial College London, for facilitating us hosting this event as well. Just a few housekeeping points, as I mentioned. Please could you mute your microphones and also switch off your cameras to help us run the event more smoothly online. And without further ado, I will hand over to Sandrine now, who will tell us more about the LCN, the Symposium series, and nanomedicine efforts. Thank you. Sandrine, the floor is yours.
Okay, thank you very much, Nazila, for kicking off this this symposium. So it really is a great pleasure to facilitate this workshop. Thank you, Nazila, for initiating the idea. At the LCN, we're also always happy to sort of support initiatives, but this one is particularly exciting. Thanks for putting together this fantastic program, also with your co-chair, Yuval Elani. So a few words as promised. So I think we have a lot of internal people here, but also externals, hopefully, who will come and join us. So the London Centre for Nanotechnology is a joint institute between Imperial College, UCL, University College London, and King's College London. So it was founded in 2005. It aims to exploit and develop nanotechnology to deliver excellent science and solutions to real-world problems, and it's organized around three main themes: so planet care, IT, and healthcare. And so obviously the last one is particularly relevant to the discussions that we're going to have today. And some of we recently sort of surveyed our community across the LCN, so our community of about over 100 PIs. And some of the challenges within healthcare that were addressed included diagnostics and monitoring, therapeutic agents and delivery, tissue engineering and implants, healthy environment, and mechanisms of life. So nanomedicine obviously fits perfectly within these remits, and this symposium is a wonderful way to share our research across the LCN community, showcase it to external audience, and hear from our keynote speakers. So the LCN has been instrumental in setting up research activities that are related to the themes today. For example, it's supported various CDTs, and most relevant to today, the CDT in smart medical imaging, joint between Imperial and King's College, and in advanced characterization of materials, joint between Imperial and UCL. So if you've got ideas today, there are ways in which you can support the research. Further particular nanomedicine efforts also include iSense, which aims to provide early warning sensing systems for infectious disease and antimicrobial resistance, which is led by Rachel McKendry, with Molly Stevens as a co-director. And also we're very fortunate to have Professor Quentin Pankhurst here, who might tell us about some of the work in nanomagnetics that are also a great stride of LCN discoveries right at the beginning of its inception. So a big role of the LCN is to connect our community, and so this is what we're doing today. I just wanted to give a word on other activities. So we've got regular informal seminars. We've got also since the start of the pandemic and the online life, we've got 1 hour joint symposium between Imperial, UCL, and King's around specific themes. The first one was COVID, but they're happening monthly, and then the half-day symposia like we're having today. We've had nanobioscience, quite recently 2D nanomaterials, and today nanomedicine. So one of the silver linings of this online life is that there is a repository, and so these LCN talks, the recent LCN talks and workshops are available on the LCN YouTube channel. Which means that, as you will have seen, today has been recorded, but so feel free to be natural here. It can be, things can be edited out, and also pay attention as if it was happening in real life, because some nuggets might be edited out as well. So I'd like to conclude by thanking you, Nazila and Yuval, and also Hafiza for setting this all up. To all our speakers, and in particular to our keynote guests as well from Silicon Valley, Omid Faroukzad and Serafim Batsoglu, and to our audience for joining us today. I really look forward to exciting science, and I hope it will stimulate further LCN collaborations and discoveries. Thank you very much.
Great, thank you very much, Sandrine. Fantastic information there. And of course, now we will be moving on to our speakers. We're bang on time. Just to mention a few logistics, each speaker has 25 minutes, after which a hand up signal will go up asking the speaker to kindly wrap up. Each speaker will then have 5 minutes for Q&A, and we kindly ask the audience to type in the questions in the chat window only in this case. Without much more, so it is a pleasure to introduce our first speaker of the afternoon, Professor Khuloud Al-Jamal from King's College London. Khuloud is Chair of Drug Delivery and Nanomedicine and Head of Medicines Development at the Institute of Pharmaceutical Science at King's College London. She has extensive experience in developing dendrimers, liposomes, quantum dots, polymers, viral vectors, chemically functionalized carbon nanotubes, and graphene oxide nanoparticles. Her current work involves development of nanomedicines for brain and cancer. She has received numerous awards to date, including the Mapplethorpe Research and Teaching Award, BBSRC New Investigator Award, the Royal Pharmaceutical Society of Great Britain Science Award, the Control Release Society Nanomedicine and Nanoscale Delivery Focus Group Young Investigator Award. She is on the management board and steering committee of the London Centre of Nanotechnology and the Children Brain Tumour Drug Delivery Consortium. She's also a member of the General Pharmaceutical Council and a fellow of the Royal Society of Chemistry. She will be giving a talk entitled Design and Nanomedicines for Brain and Cancer Targeting. Khuloud, a very warm welcome to you, and the screen is yours for sharing. Thank you.
So thank you very much, Nazila, for the extensive introduction and for organizing this very important consortium, which is the first to attend for me with the LCN umbrella on. So I echo what Sandrine said about LCN, and I can see that this is, getting into a much better position where we hoped to have it in the beginning. So let me just share my screen at the moment. My name is Khuloud. I'm a pharmacist, and I will tell you today about nanomedicine from pharmaceutical point of view and applications. You all know what nanomedicine is, but I would like to emphasize that with nanomedicine, we are emphasizing on the use of nanotechnology for medical applications. This could be an early detection of a disease, delivery of drug, or monitoring. So the all terminologies which I got to know nanomedicine with was drug delivery. Drug delivery is simply a term that we use when we want to change the pharmacokinetic of a drug. So the drug has its own PK profile distribution in the body, and by putting it in a nanocarrier, we're changing that fate, whether that could mean better activity, efficacy, reduced side effects, you name it. So nanomaterials are colloidal particles, so they have their own identity. They're not soluble molecules, and they are at the interface between, let's think of viruses as a good example. Size is important. You don't want these particles to be too small, because then they will be excreted very quickly from the body. And if they are too large, our body will recognize them as foreign particles and get rid of them. So you would like to tailor them where they have reasonable biodistribution and a plasma half-life, and they are able to find their target in the body. So these are sketches to show you how soluble or insoluble drugs could have problems, how they have very wide biodistribution in the body, and they lack targeting affinity. If you put them in a nanocarrier, the right nanocarrier, what you're doing is you're improving that specific targeting to organs, and simply you are changing the concentration in the disease target, having very low concentrations in the other organs, making your therapeutic index quite high. We work with different types of nanocarriers in our lab, and we have interest in inorganic and organic type of particles. At the moment, we have a high interest in lipid-based nanosystems, but as required, we have expertise in viral type particles, liposomes, exosomes, carbon nanotubes, graphene, hybrid materials, and so on. And really, they are our toolbox. And why do we need them? We need them because we would like to deliver different types of molecules. And these molecules, they could be small drugs, they could be radiations, they could be nucleic acid proteins, and it's not one size fits all. You need to pick the right carrier for the right drugs, because they may have better delivery capabilities or drug loading, depending on the cargo you would like to deliver. So why do we want to do this? Because we would like to target certain diseases. We have interest in cancer and in brain targeting. What's unique about what we do is we always like to image the delivery processes. So you make your drug delivery system, but before you look at efficacy or improvement in therapeutic outcomes, you would also like to confirm and perform PK studies to ensure that your drug and the carrier are getting to the site of action. So if your outcome is not what you want, that's not because you didn't manage to deliver it to the organs, but because your drug is not effective or your target is not very well validated. So this is just a scheme probably showing you maybe a bit of how I got to get to work with different types of nanomaterials in blue. Understanding processes, boxes in purple, show how actually it is important to understand how these nanomaterials could self-assemble or be formulated. So the modality aspect is quite important, and the applications are also specified here. So in terms of cancer, we are interested in cancer delivery imaging and cancer immunotherapy, and in brain delivery, we're interested in brain cancer and neurodegenerative disorders. So I'll give you 2 case studies, and I picked those case studies simply because they show the multidisciplinarity, how researchers across LCN or in the field in general are very important to produce high impact and valuable data, as opposed to us scientists working individually in silo. So this is a very good example where we have utilized carbon nanotubes to target brain, in this case, brain cancer. So a major problem we have with carbon nanomaterials is they are very insoluble in most organic and aqueous solvents. They could be made of single or multi-layers, and therefore we call them single or multi-walled nanotubes. So the first task you would like to overcome is the water solubility. So we worked with chemists who then functionalized these nanotubes with different types of functional materials to make them more water dispersible. And that chemical functionalization then allowed us to attach different molecules, biomolecules, to achieve further targeting. We then worked with image scientists and tried to really understand how these nanoparticles or nanotubes work and interact with cell membrane. And the finding we see here is how these nanotubes can directly pierce the plasma membrane of a cell, which means you're offering a cytosolic delivery, a direct cytosolic delivery to the cells, and not exploiting endocytosis, which may also lead to some degradation of the cargo. So then that also impacted on the choice of cell that we would like to transfect. So we have worked with different types of functionalization, identified what is the structural activity relationship that really matters, and the final conclusion was these nanotubes are able to deliver nucleic acids, functional nucleic acids, to different types of cells. These could be primary or saline cells. So this is just a systematic approach, how we, for example, load, in this case, siRNA to these nanoparticles simply by electrostatic interactions. You can characterize the material and ensure that you have high density of nucleic acids around the nanotubes, identify the right ratio you would like to complex them at. And we also confirm that, for example, here you can see the blue color indicates the siRNA, and when you have liposomes as a commercial transfecting reagents, you have very little internalization of the siRNA. But if you have the nanotubes, you have a widely distributed signals across the cells, which means even the uptake pattern inside the cell is very different. We confirmed if we deliver, for example, a cytotoxic siRNA to lung cancer cells, we're able to shrink these tumors. So you can see these are tumors that were untreated. You inject the carbon nanotubes as a vehicle, and if you inject the complex with the cytotoxic siRNA, you can see that these tumors shrink in size. They have a higher degree of apoptosis indicated by the green signals here, and you also have significantly lower tumor volume. This also improved the survival of these mice, which is a very important endpoint that you would like to use when you determine that this therapy is better than another therapy. So you have longer longer lifespan of these mice, or if you translate it into patients. As you may know, brain cells are very difficult to transfect, and simply that's because they don't divide, and they are very sensitive to commercial transfecting reagents. So you need to design a delivery system that can transfect neuronal cells. So in this example, we thought, let's see if what we applied in cancer can be translated into a stroke model. And in this case, we did it the other way around. So we used cytoprotective siRNA that in this case was caspase 3. So what happens in case of stroke? You have limited blood supply to a region in the brain that make some brain cells start to die, and what you would like to do is to catch that early time point post-stroke and deliver cytoprotective reagents, because the more you rescue of these brain cells as early as possible, the less damage you are going to achieve. So these are TM images that show that carbon nanotubes are able to actually internalize into neuronal cells, and they're able to deliver the cargo indicated by, in this case, the mice that were treated with a complex of siRNA nanotubes were actually able to retrieve their motor function to a pre-stroke induction level. So this is the criteria. So if you induce stroke but you treat them, actually they behave the same way as if they didn't have a stroke, and that was indicated by how fast they reach their food pellets in a rat model. Now, we want you to understand a little bit more about how these nanotubes, which are known to accumulate in liver and spleen and lung for a prolonged period of time, how can you use chemical modifications to alter their pharmacokinetic profile? So we conducted a study where we systematically increased the degree of functionalization on these nanotubes, which in effect led to improved individualization. And what we could see is the higher the degree of functionalization, the more you favor kidney excretion and retention in the body. So again, that is very useful when designing a system that you would like it to go to the kidney versus the liver and so on. As part of this work, as science is, you have serendipity playing a big role. And what we found when looking at nanotubes of different diameters, functionalized or non-functionalized with a monoclonal antibody, we found there is a high concentration of these nanotubes in the brain, which we never expected and didn't mean to look at. But the uptake in the brain was actually quite high, which is about 1.5% of the injected dose per gram in tissues. And those who work in the brain delivery field know that this is actually a big amount. So we tried to understand this a little bit further, looking at the effect of the diameter, and what we found is the narrower these nanotubes, the more they like to interact with membranes in general, so that we have high uptake in the lung and high uptake in the brain. But we wanted to take this a step further, and what we did is we applied what we call double targeting approach. So we want to target brain cancer. The blood-brain barrier is the first hurdle, but actually, if I can identify a delivery system or a targeting peptide that recognize both the blood-brain barrier and the brain cancer, then I'm able to do a double targeting approach. So we worked with the blood-brain barrier group at King's, and they had a very nice model of primary endothelial cells from porcine, which mimic the endothelial resistance of a human being. And we use a peptide called NGPEP2 that binds LRP1 receptor, which is known to be expressed both on the BBB and the brain cancer. So we prepare these particles, and this is the TM images, actually shows you how functionalization turn completely insoluble nanotubes into a relatively stable aqueous dispersion. I'm also showing you here the dimensions because I would like you to imagine how thin these endothelial cells that protect our brain are. They're very delicate. They are less than a micron in diameter, and that probably make us understand why, if you have atherosclerosis or clot in the brain, they can be easily clogged. So you have these very thin endothelial cells, and I have these nanotubes that I would like them to cross and translocate across the endothelial cells. So we functionalize them with a peptide. We also attach a chelating agent, in this case DTPA, and that is used to chelate a radioisotope like indium-111. And by doing so, we're able to do also SPECT imaging. You quantify how much of these peptides are on the nanotubes by thermogravimetric analysis, because the peptide would break down at high temperature, for example, while the nanotubes remain very stable. So these are TM images that obviously without the help of microscopists were unable to dig into this further. So again, I would like to emphasize how it is important to work with colleagues with different expertise and try to understand things a bit deeper. So you can see here, this is the endothelial cells, and these are the mitochondria. So you can see you have happy mitochondria, the cells are not dying, and the cells are recognizing these nanotubes as, you could think of them as immune cells or viruses, but they're internalizing in a controlled way into the cells. After 72 hours, they're shedding outside of the cell, and you can imagine that the layer below is actually the brain tissues themselves. So the top layer here will be the blood, and the other side will be the brain tissue. So we have a controlled process of translocation, and that was very, very nice to see. So these are actually brain capillaries taken from a mouse that have been injected with these nanotubes. So we took these mouse capillaries. You can imagine the life of the PhD who was trying actually to image these nanotubes inside the capillary endothelium, but we found them, and that was very interesting. So then the next aim was to see if I have these mice with brain tumors, and am I able to actually get more of these nanotubes into the brain cancer? So these are poor mice injected intracranially with luminescent cells, and this way we're able to see how the tumor grow without having to kill the mice, so you reduce the number of animals, and you quantify by gamma counting how much of these nanotubes are in the brain per gram of tissues, and this is after 24 hours, because you would like to get rid of anything that's in the blood, and what you detect is mostly what's in the tissues. So these are the glioma brain, and you can see the concentration of the nanotubes that is light gray, the targeted nanotubes, which is almost as twice as that without targeting, and the concentration is much higher than what you would get in a normal brain without the nanotube. So this is what actually we mean by targeting. So by targeting, you're not devoiding the rest of the body. You would still have uptake into other organs, which you cannot avoid, but you're increasing the concentration, could be twice, 3 times, 4 times into your diseased tissues. So now we are going to switch gears and talk about another type of system that we've used to target cancer. So I've always been interested in using magnetic targeting, physical magnetic targeting to target cancer. And I wondered why not many people have actually tried to apply an external magnetic field to help magnetic particles to accumulate in the tumor. And I knew a little bit why, and that's because you really like to use very small particles of what they're called super paramagnetic particles that are less than 10 nanometers, because you don't want the particles to always be magnetic. You don't want them to be very magnetic. Otherwise, they will clog the blood vessel. So you want to have these super paramagnetic, but because they are very small, they don't have very strong magnetization saturation. So it was a bit of a, how do we overcome this? So, super paramagnetic iron oxide particles are made of iron oxide. They are generally coated with hydrophilic polymer to help them circulate longer, and then generally used for MRI imaging. You can see because of the very small size, the drug loading capabilities is likely to be very low. They're restricted to drug imaging. We would like to use them in magnetic targeting, which wasn't very much explored, and hopefully by doing so, we're able to do delivery as well as imaging and maybe apply thermotherapy. So there are some limitations. The limitations are, if you use larger particles, they're very magnetic. If you use very small particles, they're super paramagnetic, and you need to make new materials which have higher magnetization saturation, but they could be more toxic, and you have to basically make new material altogether. So we had a shortcut, and what I thought we could do is actually take these small particles, build them into a bigger nanoparticles without having to chemically change the size. But by making these small ones fit into a larger one, you are artificially increasing the magnetization saturation. And the idea is, if you have a tumor which has a leaky vasculature, you inject these particles, you have the vasculature that allows the particle to extravasate by EPR effect. But when you apply the magnetic field, you prolong that retention in the tumor. So you are actually doing magnetically enhanced EPR. And we wanted to prove if this hypothesis would work. So we worked on making these nanocapsules, and there was a small trick we did, is actually, unlike what people use, we use hydrophobic particles that then will have high solubility in the oil core that then you will put inside these polymeric shell particles, and you have the polymer where you can functionalize with targeting ligands or imaging molecules and so on. You can have an optical dye inside the core, you can have radioactive probe, and you also have the spines which can be imaged by MRI. So we would like to have a multimodal system that is able of delivering drugs to cancer. So what we did, we functionalized these polymers initially, we pegylated them, we had a chelating agent on the surface, and by making a mixture of what is the recipe you want, you can get the right size particles. We couldn't get them below 200 nanometer, which I admit would have been better if they are smaller, but that's simply because of the nature of the two immiscible solvents you want to use. You can only do double emulsion method. So there is a room to improve here, but you can see they have very small polydispersity and a negative zeta potential because of the carboxylate group. So this is how they look like on the left-hand side, a cryo-TM without these spines. Inside, you can see the core shell structure, you can see the dark oil inside, and when you have the spine, they're mostly concentrated in that inside these particles. This is a 3D tomography, again showing you the geometry of these magnetic particles. We ensure that they are still super paramagnetic by doing so. And this is how it looks like. It looks like a chocolate milkshake. And if you apply a magnet, the particles concentrate, and they're reversible upon shaking, and they are also stable over time. So experimentally, we had these mice with bifocal tumors, and you apply a very small magnet that you can even get from eBay on the service of the magnet, and you apply for 1 hour. And we would like to compare how the uptake varies with and without the magnetic. So 2+ is tumor with the magnet, 2- is without. And you can see even as early as 1 hour, you have this very high concentration in the right-hand side tumor, and that difference remains with time up to 24 hours. Now, this is optical imaging. You're indirectly imaging the particles, but if I localize the polymer itself, you can also see by SPECT imaging that same pattern is observed. And this is almost 2 to 3 times increased uptake, which is a big deal. If you look at the MRI images, a darker image means you have a spine, and you can again see you have more darker regions with a 2+ compared to the non-targeted one. You can see it by staining. You can see the blue dye means you have iron oxide with tumor with targeting versus non-targeting. And to look at what do I really need? What's the magnetic field I require to have this targeting effect? So we played with things like how much iron oxide is in the particle here, and what is the strength that you apply. Consistently, what we see, the solid line is your amount in the tumor, and it's constantly higher when you apply the magnet versus no magnet at the different doses and at the different strength. You reach a limit where if you add too much of these spine, you're actually reducing the uptake. And we know now because we have more liver and spleen uptake. So there is a bit an optimum ratio that you would like to achieve. So we've done some work with Quentin, who's here today, and I mean, this has been very interesting collaboration. So we had to work with physicists, which I'm sure he can explain better than me. But what we've done here, we said, Quentin, look, we've got these images with the spine particles. Can you tell us how much forces, magnetization forces, are on each particles? And he did some modeling, and we could see there is a correlation between the content of the iron and the magnetic forces applied. Now, what is that we need? So there was a model that called the native model, and with this model you apply versus parameters, and you can predict if you are going to have magnetic targeting in a mouse or in a human by applying certain parameters. And if you fall in this region, you have magnetic targeting. If you have in a velocity region, you don't. And this is, for example, predicted in human, and you could see that there is possibility that we will see targeting.
Sorry, Khuloud, sorry to disturb. We should wrap up now if we have chance for 1 or 2 questions. Thank you.
So I'll just show you one image here where actually we can see the targeting happening. So this is a confocal images live showing these red particles that are actually accumulating as you apply the magnet, and they disappear when you remove the magnet. Okay, so we've also shown that you're able to delay the tumor growth here and improve the survival of the animal, and we can see that there is pretty much, you know, no weight loss, no toxicity, and so on. I think to wrap up, what I would say is working with interdisciplinary research is very important. You can do great stuff when you talk to each other, and obviously we can't do anything without these young researchers who spend long hours in the lab and the collaborators.
It was such a fascinating talk. It's a pleasure to see your work. And now I will open up the chat box. So if people have questions, please feel free to type in your question in the chat. And whilst people are thinking perhaps of their questions, I actually wanted to ask you a question with regards to the double targeting that you mentioned, the work you did where you had the AngioPep2, and you also had the sort of the cancer cell specific target too. And I guess my question is more related to what exactly is the mechanism? Does the particle meet the AngioPep2 first and then cross the cell and come out the other end, or was it all local targeting in the region? How does that work?
Okay, so we believe that the recycling time for the LRB1 receptor is quite high, and you have if you do the SPECT images, you see after 5 minutes actually most of the nanotubes disappear from the brain. So we believe there is a local retention in the brain in the beginning. So you have a depot effect. These nanotubes are actually sitting there. The rest is flushed, and over 72 hours you have prolonged, sustained uptake through the recycling LRB1 receptor. Mm-hmm. So this is what we think the mechanism is. And then after it gets to the tumor, everything is in the brain, everything is cleared. But because of that tumor region that has the expression of LRB1, you kind of have more interactions and retention.
Fantastic. That's very fascinating work. There is one question in the chat from Yuval Elani. He's asking, what is the manufacturing process for the CNT synthesis? Is scale-up going to be an issue where this is to be deployed clinically, for example?
So this is a very good point, Yuval. So what we noticed is actually is not that you only have to stick to one supplier, you need to stick to a batch when making these particles. So we weren't synthesizing them, but we were functionalizing them. So when you buy these ones, for example, we identified a couple of suppliers who have less metal contaminations in them. Obviously, we would have acid treatment to clean them, etc. I mean, this work has been done quite some time. I'm sure there is more QC in the field of the synthesis, but you've raised a very good point that the variability between manufacturers is vast at the moment.
Great. Okay, if there are no other questions in the chat and from the audience, we will thank you, Khuloud, for a lovely presentation, very interesting insights into the world of release carbon nanotubes. We'll move on to the next speaker. Thank you very much, Khuloud.
Thank you very much.
Thank you. And so next up, we have Dr. Giuseppe Battaglia, or Beppe, as everyone calls him. Dr. Battaglia holds the double appointment as Chair of Molecular Bionics in the Department of Chemistry and the Institute of Physics of Living Systems at the University College London, and as ICREA Professor and Senior Group Leader at the Institute of Bioengineering of Catalonia, part of the Barcelona Institute of Science and Technology. Beppe holds a laureate in chemical engineering from the University of Palermo and a PhD in physical chemistry from the University of Sheffield. Prior to this current position, Beppe held positions as lecturer in the Department of Material Science and Engineering at the Croter Institute and senior lecturer and professor in the Krebs Institute and Department of Biomedical Science at the University of Sheffield. Beppe's research involves biological transport at multiple scales. His group combines physics and biology to elucidate the relationship between macromolecular structure and trafficking and how these move across tissues and cells. They also use this information for molecular engineering of novel nanomedicines, creating unique solutions for precision targeting. Today, his talk is going to be entitled Phenotypic Nanomedicines to the Brain. Very well, welcome to you, Beppe, and the floor is yours for sharing. Thank you.
Thank you very much for the kind introduction. Next time, I promise you a much shorter biography.
That's fine, thank you.
Do you see my screen?
We can see your screen, yes, thank you.
So good. So it's great to be here, and it's great to speak to the London Centre of Nanotechnology, which is actually in front of my office, and it's been sitting, at least the UCL side, for many years. And strange enough, I don't know why I haven't really interacted as much as I should have, but hopefully things might change in the future. However, there is a change in my geography because my group is gradually shifting a lot of its activity down south. I think it was time for me to emigrate a little bit south, or getting a little bit nostalgic of the seaside. So although what I'm presenting to you will be mostly based on results that we've done at UCL, gradually my group is shifting towards Barcelona, where I will be joining. I've joined already in the last 2 years the IBEC, the Institute of Engineering of Catalonia. My research group is indeed a balance between interdisciplinary. I'm very keen to maintain that balance as much as possible. We do have a bunch of physicists, a bunch of chemists, and a bunch of biology working together. And this is really creating a great atmosphere for fostering discovery. Of course, today they all talk about is the nanomedicine, although we have activities in physical biology, molecular engineering, and biomicroscopy as well. Of course, I will try to cover here most of our activity in nanomedicine. And we have effectively branched out across many different clinical areas in immunology, in oncology, and neurology, and mostly to do with the fact we're actually developing a platform technology, which is now also being spun out into a company called SomaServe, which is a UCL spin-off that has been operating for about 2 years or so. This is kind of a disclaimer. We're also trying to spin out one of the products out of this technology into another company called SomaNautis for brain cancer nanomedicine, but we're much earlier stages on that side. The two platforms which are really critical for us was intracellular delivery and phenotypic targeting. And today I will cover as much as possible phenotypic targeting. However, bear in mind that whatever we do always come with the ability of intracellular delivery. So what I want to focus the attention today is on personalization, and how important it is to really make sure that we can address and make sure our medicine treat patients individually. And this, of course, has been possible thanks to the revolution in the omics in biology. We're now literally mapping out individuals, disease states, part of the organism, down to the single molecular composition and macromolecular composition, and discovering that actually there are differences between patients, and every cell has its own phenotype, i.e. the composition of the receptors there. So how can we make sure that our therapy becomes indeed capable of recognizing these differences in phenotypes and adapt it into something that becomes extremely precise? Now, what we do in our labs, we combine both computational biophysics and some computational science, but very basic one and still very early stages, with experimental sciences. In particular, the idea is to really develop tools that feeds from information from bioinformatics. So in other words, what is the composition we want to target, and then through a correlative and cyclic optimization loops, we create a system that really indeed allow us to identify phenotype from phenotypes. We are not there yet, but what I'm going to tell you is where we are at right now, and hopefully you're going to believe that we are quite in a good place, and hopefully maybe in a few years' time you will see some more upgrade about this story. What I'm going to do, I'm going to focus, first of all, on the general concept of selectivity and targeting. This is a big challenge, and indeed one of the most difficult ones to achieve in medicine. And when I mean targeting and I mean selectivity, I mean the ability of a certain molecules or macromolecules or nanoparticles to reach a specific part of the body. And most importantly, reaching that part of the body which is targeting has to happen with the highest selectivity possible, which is on the other hand is to make sure that that interaction is focused and localized in that part alone. This picture that you see in this video is actually something I stolen from the internet, and is a CT PET scan of a human being with a brain tumor. And you see the brain tumor is very much isolated in a particular part of the brain, and indeed has a very, very specific characteristic, metabolic profile, marker, etc. And the typical approach in medicinal chemistry to identify a marker associated with tumors, really getting down to the molecular level of the marker, passing through tissue, the histology, and the cellular levels, and then really understand down to the molecular details how the proteins of the marker that characterize that disease operate, etc. And that information is critical because we can quickly transform it into medicine discovery. You can, of course, bypass all of the drug discovery by simply creating a genetic engineering route where effectively you intervene in that biomarker by delivering nucleic acid, and that transform the genetic codes of the host to really transform the function of that biomarker. And that is, of course, the gene therapy. But traditionally, what we prefer to do in medicine is to create molecules that bind and stick to that marker and do something to it. Changing this function or perhaps inducing some sort of signaling process. And typically, depending whether we use antibody or we use peptides or we use small molecule, we always screen and try to identify the molecule that binds the strongest to that marker. This process is called technically as high affinity discovery process, and indeed has been driving the medicinal chemistry for many, many centuries. Actually, as long as medicinal chemistry ever existed, and of course, I don't know many of you, the father of medicinal chemistry was this German scientist called Paul Ehrlich, who postulated in Latin then corpora non angunt nisi ligata, which means the substance is not effective unless it's linked to another. And indeed, it was the father of side chain theory. It was the person who coniated the terms ligands and receptor. Indeed, he came up with the idea of really selectively targeting things chemically, really introducing the idea of modifying molecules that really interact with high specificity. Now, this concept really now built up into a much more advanced system, but what I want to do is really point out some sort of interesting paradox that leads to problems. Because if we design a molecule that binds the strongest to the target, yes, you're going to get an interaction to the point where you have the highest concentration of your target, let's say our brain tumor here, but you also inevitably interact with molecules and cells that have also a little bit of that receptor, so something even worse, have a very similar value of our disease state. Yes, you avoid other interaction and more specific interaction, but you're still left with a problem of selectivity there. And indeed, this problem becomes quite critical once we look at the whole body. So if our receptors, our target is expressing in brain tumor, very often this is not uniquely expressed. And typically you're finding in many other parts of the body. Actually, the receptor in question I'm showing you, LRP1, is indeed expressed in very high level into fat, liver, lungs, etc. So indeed, our high affinity means that not only we're going to interact with a brain tumor, but we're going to interact with a lot of things. Our targeting is not selective. And that means, of course, very quickly and very easily side effect. And indeed, these can be quite dramatic when it comes to oncology, where you want to kill your cancer targeting, you want to transform targeting into killing. And indeed, if you, instead of killing cancer cells, you start killing healthy cells, you understand the side effect can be quite damning. And as a matter of fact, clinical trials have been failing one after the one, because most of the time, it's particularly for this problem. Efficacy, mostly to do with the fact that you don't really reach your target, or you don't reach it with high selectivity, and safety, where you, of course, you lose your targeting into side effect can be very detrimental. As a matter of fact, in 2016, the average likelihood of approval of a drug going through clinical trials was below 9%. In an area like oncology, that number goes down to 5%. So what's going on there? Let's try to break down the problem in physics terms. When we have an interaction between a ligand and a receptor, this is very much can be simplified as a reversible reaction. And effectively, a reaction that goes to a rate of binding and rate of unbinding. And indeed, the binding energy or the formation of the complex can be written down as a simple equation. Now, if I take these concept and apply into a cell, so where I either have a cell with one receptor, let's say a cell with 3 receptors, which would be our target, of course, my possibility of creating a bound state with the cells with higher receptor is higher. So in other words, the output number is 3, our omega here. And indeed, our equation becomes a little bit more complicated. You start to add this sort of number of receptor into your equation. And indeed, if we plot the energy of binding, I'm going to use KT, and I'm going to put our 0 being between 0 in terms of fluctuation, all the way up to weak binding, which is basically water-hydrogen bonds. And then we use as an x-axis the number of receptors into a target into cells. So with our target cell being on the high numbers and other cells in there. If we use a high affinity ligands, and naturally those numbers I'm putting there are not that high at all, you see that though it increases with the number of receptors, the energy of binding is sufficiently strong to get interaction even when you have only one receptor. You don't really get any selectivity whatsoever. Now, you can get a little bit better selectivity if you go down to affinity, which are very much impossible and almost too weak to actually create a binding system. That's just to say, actually, if you want to screen for selectivity, the high affinity is not the right protocol. Now, things become a little bit more interesting if instead of using a monovalent system, we go for a multivalent system. Just for the sake of argument, I use a trivalent system. And this can go, instead of the number of output already multiplied by the number of valencies. So one receptor will have a 3. And of course, when I have 3 ligands targeting 3 receptors, this can go up to 9. This number can become a little bit bigger depending how you express those ligands. In other words, what's the topology of your binding, how much space every ligand has got to explore the rest, etc. Without going too much into complicated maths, you can see that this number can change even order of magnitude in terms of increase. And that means that our energy now is no longer changing so linearly with the number of receptors, and indeed we start to see a non-linear trend. Now, if we apply this to our high affinity, we still don't get much selectivity. Yes, we have an increase of energy here, but we still bind very much with the one receptor. But on the other hand, if we start to use the bad affinity system, and we put it in a multivalent, we start to observe an energy profile where you have a very, very weak interaction at very low number of receptors. In other words, we start to create a system where you have a non-interaction, a very weak interaction below a certain threshold of receptor number. Now, a better mathematical way to represent this is using a parameter called theta, which is the fraction of bound particles, which goes from 0, non-interaction, 1 interaction. And indeed, our high affinity system are again pretty much useless as selective cells, and they always bind independent whether you have one receptor or 100 receptor. On the other hand, if we get our really weak ligands, there is no interaction whatsoever. Like I say, the interaction are too weak to actually saturate our surface. But on the other hand, if we combine them multivalently, already with 3 ligands, we see this beautiful on-off binding profile, where again, we see this sort of onset where everything starts to happen. And most importantly, this process is very highly nonlinear, and this law really critically makes a big difference in how things happen. Now, However, in the real system, it's never really one ligand or receptor. So your receptor is immersed in a matrices of really complex, high complexity. A cell membranes got tons of different type of receptors. It's got a lot of other actors like sugars, glycans, and etc. All of them moving very, very dynamically and inevitably interacting with each other. So our binding energy is never really a single entity or a single event. It always has to deal with the presence of other molecules. And this can often create a steric effect, indeed creating repulsion, whether that comes in the form of a next receptor or a long glycan chain, or whether that comes in the form that part of our ligands actually is different and indeed doesn't like the receptor, or whether our polymeric ligands can stick in one part, of course, in its binding site, but the rest of the receptor doesn't like the ligand at all. So all of this creates an interesting scenario where the energy actually is a combination of positive repulsions and negative attraction. And indeed, if we start to look at this again with our diagrams of energy, what we see here is an introduction of a new component, which comes from the stereo, which we can approximate to be linear. That means our total energy has now a non-monotonic behavior. And again, if we transform this into theta, with this subtle, really beautiful range selectivity, effectively our binding occurs in a very, very narrow range. And this is just simply to do with the fact you have a combination interaction. So in all effect, we have a tug of war of interaction. And indeed, what we're trying to do is really mathematize all of this process into some sort of working principle that we can use for our design. So that we can go back into our cells, we of course try to understand how the binding happens, and we can use molecular dynamics and molecular docking mechanisms to help us out. We have a lot of experimental validation, but we also try to identify things which are associated with esterics like ligands, etc., in particular protoglycans and glycoproteins, which are critical. So with this in mind, how can we create this surface complementarity? How can we make our nanoparticle interacting only with that specific phenotype? So indeed, what we do here, we start by building a material which is really compatible with the human body. We use a materials called polymersomes for many, many years. I did my PhD on those systems. It's a synthetic vesicle which formed by the self-assembling of block of polymer in water. And what we do here is making sure that our chemistry is very safe. In other words, we choose exclusively material either that are part of the GRAS list, so generally recognized as safe, or materials that we know our body can metabolize. We then create from those ingredients a functional multivalence and multifunctional nanoparticles. And then what we want to achieve here is that after it reaches its site and deliver whatever cargo we encapsulated, then the ingredient degrades and are really weakly metabolized in our body. The polymersomes are extremely versatile in controlling many different molecular engineering parameters, from tunable mechanical properties to ligand functionalization to self-assembly. There are now many different ways to make them in a controlled form and shape, but they're extremely stable and they're really pretty good for biological application. In particular, one of the critical elements that for us is very, very important is the ability to withstand a highly crowded environment. Because when you go into a biological fluid, you're never really just in water, you're actually surrounded by a lot of proteins. This simulation, which I show you, which I created with cell paint myself, is effectively a simulation that accounts for the typical protein encounter in the plasma in our blood. The gray one are albumin, you can recognize the IgG, then you can recognize the IgM, fibronectin, and lipoprotein, etc. Now, the amount of protein I put in this simulation is exactly what you would expect. 10% of volume fraction of our plasma is actually occupied by those proteins. Indeed, our nanoparticle inevitably will encounter those proteins. So the very first rules that we need to apply in our design is making sure that these are not sticking to the surface of our nanoparticle. Otherwise, everything is controlled by those proteins and the interaction with those proteins. Our ligands become completely useless. And indeed, this is possible by creating the right chemistry. You probably know PEG as the most famous of all, but most importantly, I'm not going to go too much in detail, the physics. You need to create a very, very dense brush. Just a simple PEG chain is not sufficient to actually stop the interaction. In our case, our polymers are pretty very good, that you can get 1 protein every hour sticking. It's impossible to stop completely protein corona, but it's good enough for us to actually create a system which is inert. And then it's just matter of chemistry. You can attach whatever ligands on the surface. So what I'm showing here is one type of PEG where it's very linear, and the other case is where it's actually branched, poegma. And then we use many different clicking and conjugation strategies. And the idea is to create a system that has either the ligand inserted inside our brush or the ligand surrounded by a very, very dense brush, although short. So these two are quite important because both of them are very stable and ultra low protein fouling, but most important, allow us to really control that interaction I was telling you, introducing even more nonlinearity into the system. Because now for our ligand to reach its receptors means that the receptor needs to enter the brush. Effectively means that our binding again is controlled by our proper molecular interaction plus the steric potential. This allows us really to create a very, very nonlinear behavior. And most importantly, if we multiply our receptor types in terms of type of receptor we're targeting, what I call multiplexing, this effectively allows us to really create a barcode in our targeting capability and really isolating cells in terms of the phenotypic composition, not whether they have or not their receptor, but how much of those receptors and what's the relative composition of those is there. So we apply this in our design, and indeed we're creating a mathematical model that allow us really to account all the different components, both the active interaction, both at the molecular level, as well as the sterics, recognizing different components, not only exclusively by the polymer we introduce, but also by the GAGs, the glycocalysis surfaces around cells, or even the single glycan that surround the receptor in some cases. Now, these models then feeds into our loop. What we do then next is to really validate the model. Of course, this was already done. And for that, we need some sort of really cool biological application. I don't need to say much about the blood-brain barrier. Khuloud has done a great job for me already. But I want to point out that the BBB is effectively the vasculature that feeds into our brain, and effectively is made by cells, which are the same cell that made any other vasculature in our body. The only difference is in a brain, the vasculature gets reprogrammed by the surrounding cell, by the CNS cells, creating a very unique phenotype associated with that BBB, but still very much an endothelium phenotype. And this is really important. So the differences of targeted BBB and a non-BBB endophilia is not that trivial. There are very, very, very small differences in some cases. So indeed, we apply our systems, and of course, the very first part is really bioinformatically understand what receptor you want to target. We discover the LRP1 is highly expressed in the brain at protein levels. We discover the other receptor also very interesting, GLUT1, EGFR, etc. Also, we identify receptors associated with trafficking. I'll come back to those in a second. And most important, remember those sterics here representing the forms of protoglycans, which are highly expressed in endothelial cells. Again, we put them together, we understand the ligand topology, we have our chemistry, we identify binding, and indeed, after a lot of screening, instead we create this sort of binding curve where you have either the actual fluorescence per cells, or you normalize using the theta parameter. Remember that 0 to 1 means 0 no interaction, 1 interaction. And indeed, we identify our theory works very well and fit our data extremely well. And we also show this sort of beautiful synergistic binding where you combine multiple receptors to target that particular phenotype. Now, what really is beautiful is that everything is designed at the molecular level. So by changing the molecular arrangement, in other words, those sterics potential, not the binding is still the same. In this case, I'm showing LRP1 bounded with angiopeptide, which was introduced already by Khuloud, the same ligands here. The only difference here, what I'm showing is basically the way we present the ligands and how many ligands we have per pathway. You change completely the way that these interact with the brain endothelial cells in there. Now, one thing is binding, the other thing is getting across, and that of course means understanding how endothelial biology works. We spend a lot of energy in that. We use a very similar in vitro model that Khuloud already introduced. What we did here is screening for the particles that get across with the different type of ligands. And then particularly, we complement those with in silico simulation using particularly agent-based modeling or other tools. And indeed, what it appears, again, is non-linear nature, even more so when we introduce the trafficking site, because not only you need that specific avidity or energy of binding that in this case is represented by dissociation constant, but also you need to remember that you need to have enough energy to bind in one side, but also as you get across, you need to have energy to unbind on the other side, because if you get bound with high energy, you actually get processed in a very different way. And indeed, what we discovered recently is that this really makes a big difference in endothelial cells, and endothelial cells transfer materials, really controlling things through these avidity games. And mid-avidity actually get processed in a tubulated trafficking vesicles, which I show you by super resolution state microscopy in real time, moving and forming. This is the apical and this is the vesilator, and this is an ex vivo snapshot of a capillary walls where it shows how the polymersomes can get across, and particularly associated with this protein syndapin2, which is giving a lot of joy in order to mark down how BBB works. And then finally, of course, in vivo, this nonlinearity really emerged even more so, and indeed, you actually so much dependent on these very little changes in very narrow input that you need to be really, really careful as you look at your biological data in general. What I'm showing here, that actually we get some very good brain delivery with 22 units of ligands already. When you have a 40, you don't get the same, and with 100 even more so. So actually, the more is not the best. Is not the more in this particular case. Now, what we can do with this, of course, the delivering drug is the very first things. This is one of probably the best data we ever got in our group. We were able to encapsulate the platinum compounds, which is not only very bright and therefore visible in many different fluorescence imaging techniques, but also the platinum allows you to characterize everything by mass spec. And so we did that. And indeed, what I show this graph is that our particles that target the brain can deliver the large majority of the drug in just half an hour after IV into the brain. And indeed, the other organs have really little touch. After 24 hours, you still have interaction. This is for another lectures. I will one day I will show you how dosing works. This is very important when it comes to nanomedicine. But most importantly, more clinical application. This is a collaboration we've done in collaboration with the group in Sheffield, the Arshad Majid, the Oknam Bae at the Hagyan University in Korea, where they were interested in delivering a neuroprotective agent for stroke. They had a very good stroke models, and indeed we showed that we can downgrade the dose of the drug of 1,000 times just because we deliver the stuff into the brain, and that exactly the neuroprotective effect we wanted. Now, a little bit more sophisticated targeting. This idea of phenotypic targeting becomes even more so if you want to do double targeting. In particular, we started big programs in glioma, and now we apply the same principle here. We map out the receptors on endothelia, brain endothelius, and glioma. Turns out that some of these receptors, particularly in glioblastomas, are highly expressed. And actually, LRP1, the same receptor we use for crossing, is also highly expressed into tumor cells. So that really helps out a lot in our first prototype. And indeed, we've been doing this also, this mapping out of the vasculature locally. And that's very important because the glioma changes the way that the vasculature phenotype adapt. Indeed, we see huge changes in both receptors, glycocalysis, and transport property in particulars. And what we're now doing, we're adapting this information to really adapt our targeting. And what we found out, why the formulation that works very well for healthy animals also works for tumor, as you see in this graph here on the right, we found out that actually increasing the ligands number makes even better way of delivering to the tumors. And it looks like actually the healthy one works through getting into the tumor from the healthy vasculature of the brain, while the one a high number of ligands, which is we only found in the tumor, only gets into the tumor vasculature. And that's beautiful because it allows us to target the tumor from different direction, which is from a phenotypic at the tumor level also makes a big difference. And indeed, this means you can also do this. I'm just showing this that it's not only by IV, we can do IP. This is important for pediatric model where you work with little pups. But what really means this is really high efficiencies in really getting the drug inside your cells. I already mentioned at the very beginning we do intracellular delivery. Our system already in vitro shows some huge enhancement by combining intracellular delivery with delivery, because we escape endosome using pH-sensitive system. And most importantly, in vivo, we see some very interesting, really promising data using animal models, genetically modified animal models, which have glioblastoma extremely aggressive, which are very difficult to actually cure. And indeed, we see pharmacological effects such as apoptosis emerging only exclusively in the system that we target. With that, I finish. Of course, I need to acknowledge a lot of people that made this possible. This work was done mostly, the work presented by Diana Leite, Diana Matias, in my group in London. There are also groups that are now catching up on this, Daniel and Katia and Rui will continue on the glioma, Belen on the drug, Edo to the left. And of course, our collaborator, I forgot to mention Simona Parinello from the Cancer Institute for the Glioma, and Sebastian Brander for this neurology as well, for the animal models for the glioblastoma. And then, of course, my sponsor, in particular, the ERC and APSRC are the major, and the Cancer Research UK and the Children with Cancer UK are the four sponsors of the data that I show you today. Thank you very much for your attention. I hope I didn't do over time.
Thank you very much for a really fascinating talk indeed. It's really fantastic to see such a deep level of insight into targeting and matched with nanoparticles as well. I mean, I for sure have a few questions, but I will now open it up to the audience. Are there any questions for Beppe, please?
I'm gonna stop sharing meanwhile because I think I don't see many people. Yes, better.
Yes. So there is one hand up. Is that your hand, Lioval? Please go ahead.
Yeah, great talk, Beppe. I love the physics bit at the beginning. It's always nice to see the physical underpinnings of all this. My question is about the polymersomes. You know, they're really fascinating. You know, I'm more used to working with lipid systems, but polymersomes clearly have more chemical diversity, and you can see why they can be very attractive. So I guess my question is, you know, if you look at sort of, you know, the field in general, people tend to just first thing go to lipids, liposomes. You know, is there a reason that, you know, people aren't, you know, that polymersomes haven't surpassed liposomes in terms of number? Is it just an inertia thing that people just tend to want to stick with what they know, and, you know, or is there something more inherent? Is there an inherent difficulty in working with polymersomes?
No, absolutely. There is an inherent difficulty working with polymersome, and indeed there is also an historical delay. I mean, liposome were discovered in the 60s, the polymersome were first reported in the late 90s, right? So there is a bit of a 30 years advantage that the liposome have over polymersome. Also, the preparation of vesicles is not as simple as with liposome. Polymers tend to a little bit more temperamental, so you need to really use different methodology, etc. They're getting more and more popular. I think when I start working on it, I mean, I had to really use anything from liposome community and adapt them, and not all of them worked. And unfortunately, we never really published our bad data. We probably should do that. But indeed, they are becoming more and more popular. Of course, the raw materials are so critical. You know, we have chemists that make our polymer in our group. Not always you find a raw material in Sigma Aldrich or polymer source, so you need to have a chemist collaborator. So why phospholipid, again, you go Avanti, right? You buy them in tons now, particularly with the vaccine boost, right? But what I'm saying to you, I think there is a delay. They definitely, the advantages in terms of vesicular morphology are equal. The advantage of polymers, of course, is the fact we have a very, very thick brush and much, much bigger control on the molecular level, but at the physical level, they are soft, they're beautiful, they interact very well with cells, so you can use entirely grass materials which are already approved in the clinics as ingredients, and still you get the same thing.
Fantastic. We also have a talk from Maya Thanu. Question, sorry.
Okay, great. Thank you very much for this fascinating talk. Of course, I mean, it's exciting to see these cancer drugs. Particularly, I'm very glad that you're using cisplatin, and it's very easy to be detected. ICP-MS, TXRF as well. It's easier and cheaper. So just comment. But I just noticed that your polymersomes with cisplatin were all over the brain.
Actually, it wasn't cisplatin, Maria. We actually use platinum compounds.
Platinum, yeah.
Parts of it is similar to cisplatin, but the other part has a very big fluorescence. And the way we use it, because it's a platinum compound, so you can sell it as an anti-cancer drug, but also it's extremely bright. So you actually you can detect really almost a single molecule levels, and that really allow us to know where we have gone through. And indeed, we go through all the whole brain. We see stuff all over the brain.
That's absolutely.
And that's why, you know, in the next part, we are optimizing the targeting towards the glioma vasculature to avoid exactly that. Because with cytotoxic, although the brain is not such much, I'm sorry, anticipating your question, right?
That was my question.
But basically, that's true. Actually, I got really scared when I saw those pictures, Maria, to be honest. And you should see the histology. If you want, I can show you. It's everywhere.
Everywhere.
And indeed,
Yeah.
I got really freaked out because we're talking about cytotoxics, and indeed, now we are a little bit more confident.
Okay, because this is my question. I mean, if you have platinum compounds of whatever for therapeutic reason, even if the polymers, they go everywhere, how can you guide them, like to the tumor? So, and I'm expecting to see the solution. And of course, I mean, all these polymers that you provided, that might be better for other diseases, brain diseases.
Sure.
But I think for the brain, for glioma, for
I absolutely
glioblastoma,
agree.
yeah, it must maybe better, particularly if we deliver the cytotoxic, to go target it.
Absolutely. To be honest, in our data with doxorubicin, which is not as toxic as platinum, we don't see much toxicity in the brain, or the apoptosis is very much almost limited to the glioma area, in the limitative area, right? However, it's a risk we need to take, and we need to see how it goes, right? But I agree with you, and indeed, our ability of tuning the targeting, in particular when we added the double targeting, so combining LRP1 to get across with something like EGFR or any other targeting modalities that we are using for glioma, allows to really homing things into the tumor.
Fantastic, particularly if the brain tumors from children. I mean, you can't deliver anything else at the healthy brain. I mean, you have really to target on that. Thank you so much.
So those animal models, actually, Maria, were really, really young pups. So this is that's why we really care about the toxicity. Thank you for the question. That was really good. Thank you.
Sure.
Thank you very much, Maya. If there are no other questions, we should thank Beppe for an excellent talk again. Thank you very much. And moving on to our next speaker. Thank you.
Thank you.
Thank you. So we're a few minutes behind. However, and the last speaker for our session today is Professor Quentin Parkhurst. Quentin Parkhurst is Professor of Physics in the Department of Medical Physics and Biomedical Engineering at UCL. He is also Director of the UCL Healthcare Biomagnetics Laboratory, which is situated in the Royal Institute of Great Britain in Mayfair. Quentin Pankhurst has a long time association with the LCN in Bloomsbury, having been involved in its inception and acting as its Deputy Director from 2002 to 2008. And Quentin, we are delighted to have you here today, and we welcome you to present your talk and share your screen. Thank you very much.
Thank you very much, Nazila. Let me show you my... Am I allowed to share the screen? Yes. Okay. So can you see that now?
Yes, we can see your slides. Thank you.
Okay, so thank you very, very much. It's a great pleasure to be speaking to you at the LCN. I was looking in my notes, and it was 2007 the last time I gave a talk at the LCN. So I must have done something wrong. So, but I'm back now. Long time no see. I'm going to talk to you a bit about biomedical applications of magnetic nanoparticles. Not exactly nanomedicine. It's a combination of medical devices using nanotechnology and also combination devices. But most of the things I'll be talking to you about today are focused around nanoparticles as medical devices. So as mentioned, I'm in biomedical engineering at UCL. Healthcare Biomagnetics Lab. And I'm also going to tell you a little bit about some work I've been doing with 3 spin-out companies. And I'll start the talk with that. But even before starting that, but just to refer back to something which Khuloud had mentioned, which was that magnetic nanoparticles have been used for decades in clinical applications, most notably in medical, in MRI, where they are used for contrast agents. So you have an image here of somebody holding a vial with a magnetic fluid in it, and what's inside here are magnetic particles which are really rather small, 5 to 10 nanometers in diameter, encapsulated in a sugar or a starch, with a hydrogen bonding between the surface of the iron oxide and the sugar or the starch. This is showing a diagram for dextran. And they're used for contrast agents. On the left here, we have an image of a lesion. This is a cancer tumor in the liver in this patient. And on the right, you see the same image, but with slightly better definition of the margins of the tumor. And this is because the contrast agent shows up as a dark image under T2 imaging. And this is an improvement for surgical resection where you can identify the margins a little bit better. So magnetic nanoparticles have been used for decades. They were introduced into use long before the concept of nanotoxicology became very common. So that we have the advantage with these particular types of iron oxide magnetic particles that they have a long and established clinical use history. They have a size which is useful from a biomedical perspective. They have other properties which I'll be highlighting today. One is that they can be actuated by a magnetic field gradient, and secondly that they can be made to heat up. So I'm going to be talking about applications in magnetic sensing, in heating, and magnetic actuation or moving. The first of these, I'm going to highlight the work done by the three spin-out companies that I work with. The first is in magnetic sensing, colleagues at UCL and now at Guy's. Actually, no, Michael Dueck has now moved up to Oxford. And the concept here is the staging of the treatment of breast cancer. So the principle here is that when you have a primary tumor in the breast, it becomes important for the staging of the treatment to know whether or not the cancer has spread into the lymph nodes in the armpit. And for this purpose, one injects a tracer around the nipple or around the tumor and look to see the lymph nodes that the lymph drains to. This may work. This is a video showing one of the very early operations that we performed at UCL Hospital, where we are using a handheld magnetometer. It's actually a susceptometer to trace the location of the magnetic particles which are in the armpit here. That's where the lymph nodes are. Up here around the nipple, you can see where the injection site was. So the principle there is that you get both a visual and a magnetic signal from the node, and you're able to identify it and take it out. That's where the actual staging of the treatment takes place, is to determine whether or not the cancer has spread, whether you have metastases in that node or not. We've actually introduced this to Europe in 2012 and into the United States now. It's actually quite a large number of patients have received this treatment, 65,000 people to date. And I think it's starting to become one of the standards of care for sentinel lymph node detection. Very good concordance and correspondence with the existing method, which was a radioactive isotope-based one. We are also now working with a different type of application using the same detector. This is a seed for lesion localization. And that's also now in the United States and also coming up to 50,000 patients. I'd like to say a little bit about magnetic heating or magnetic hypothermia. This is work done with Paul Sutherland and Simon Hattersley in the company Resonance Circuits Limited. And the principle here is really rather straightforward. If you think about a magnetic hysteresis loop, as you pass around the hysteresis loop, you do work. And that work is translated into energy in the form of heat. Another way of thinking of it is that we're looking at the magnetic field equivalent of the microwave oven, or you could say another way of thinking about it is that it's like an induction heating hob in your kitchen. But the principle is very straightforward. We take a solution of magnetic particles in water for injection. We inject it into a tumor, so direct injection in this place. People are very excited about the idea of having intravenous injection and targeting using antibodies on the surface. But we're much earlier than that. We haven't got there yet. We're trying to prove the direct injection route. into the tumor, healthy tissue around, and then when you turn on a time-varying magnetic field, it heats up the particles. This is an example. Which I'll turn.
And we've injected the nanoparticles in the center.
My colleague Paul Southern here is showing a demonstration using the particles.
The area which has the magnetic particles will only heat up.
And you can't feel anything on your hand then?
Nothing at all. So there's no tingling sensation whatsoever. The exciting thing about that is that these particles are really small, much smaller than a cell. So
it's a bit awful to hear yourself on a video. So here we have the hot spot that we've managed to generate, and the idea is that you do that inside the body at the tumor, and that you use that heat as an adjuvant, or as an additive adjuvant to other treatments like radiotherapy or chemotherapy, or as a standalone treatment. One of the difficulties we have actually is that there are so many possibilities. We've taken this quite a long way now. This is an image of the base unit that we have, and this is actually at the end of last year. This is a base unit which has been packaged up, just about to be sent off to Barcelona, where we are currently recruiting. We haven't yet had our first patient, but we're currently recruiting a clinical study for treating pancreatic cancer. And the material that we're using as the heating medium is a magnetic fluid, which has got very good magnetic heating characteristics. One of the things that we have to worry about a lot here is simply the production of sterile materials for injection into the body. A third area that we're working is with magnetic actuation. And this is with a company called Medicine, and with some colleagues of mine, George Frodsham, Christina Blankenduja, and Will Twigger, and their colleagues in this company. And the principle here is really also quite a nice one, I think. It's quite straightforward and simple. It's using, so Khuloud was talking earlier about magnetic actuation of drugs to cross the blood-brain barrier and to reach other difficult targets. This is a slightly simpler principle where we're trying to remove pathogens from blood by using a magnetic particle, which is functionalized with antibodies or other targeting moieties for cells or toxins or cytokines or whatever. And the principle is to have an extracorporeal circuit through which we pass the beads and then take them out again. So in a sense, we only mix for a teeny weeny part of the circuit here, but the idea is to mix long enough to be able to pull out our target toxins. Now, we actually were fortunate enough to get a reasonably big grant from the government for using this to treat COVID-19. And the reason for that is that one of the key cytokines which causes the most distress and damage to patients during COVID-19 is the same as one that appears during sepsis, and that's interleukin-6. And one of the things that we're trying to do is we can't stop the IL-6 production completely, but what we can hope to do is to reduce the severity of the infection and improve the body's ability to recover. So instead of going through to the systemic and inflammation, the tissue damage, and the multiple organ failure and death stage, to bring us back to a recovery stage. We've done quite a lot of work on this. This is the data taken from last year. We actually have a human clinical study proposal at the MHRA at the moment, which we're hoping to get, as soon as we get that approved, we'll be able to do our first safety tests with this. And our goal is to be actually getting into clinical studies at in midway through next year. But we're looking at all sorts of ways of optimizing what is already a reasonably good capacity for pulling out particles with our magnetic filter. This is just an image from a GLP animal study that we did a couple of years ago, proving that we're not going to be upsetting the blood chemistry specifically. And so this is a test of whether or not there were any adverse events associated with that GLP study, which there were not. And this is our performance data in vitro benchtop studies, where these two graphs are essentially we're spanning the levels of IL-6 that you see in sepsis and in COVID-19, and showing that we can get on the benchtop, we can remove pretty much 90% of the IL-6 in 8 or 10 minutes. That corresponds in a human, which is a much bigger volume, of course, and you get a lot more mixing going on, to a reduction of about 80% in about, was it 240 minutes? It was, no, 180 minutes, 3 hours. So that's our target. Okay, so can I, Nazila, if you're there, can you just give me a time check? How much time have I got?
Yes, absolutely. You are 12 or 13 minutes in your talk.
Okay, I'm going to try to finish early so that we can have a bit more time for conversation. But what I'd just like to say a little bit now is that I've said a bit about the spin-out companies that we've managed to work with, but there is much more to it than that. And within, there's a lot of work goes on behind the scenes to get towards clinical translation. And I just want to touch on 3 different aspects of that. I'm going to do it relatively quickly. We've been working with people in CABI at UCL on in vivo tracking using a process we call surface radiomineralization. I know something that I'm so sorry, I've forgotten his name, but there's a very good group on this at King's, but this is a slightly different way of essentially taking our MRI detectable iron oxide nanoparticles and surface treating them in a way which produces a mineral, a radio mineral, MX06 shell on top of the particle, more like islands, not shells. And we've been doing this with both indium with a half-life of 67 hours and zirconium with a half-life of 3.3 days. And the point here is that we want to have a long-lived half-life on a radioisotope so that we can actually track and follow the way in which the materials in which we are wanting to clinically introduce into the body, how they are distributed, and also, most importantly, how they are then metabolized or excreted. So we have some HRT-EM data here showing which we used to satisfy ourselves that we were indeed seeing surface mineralization, similarly with some Mossbauer spectroscopy, similarly with EDS and TOS SIMS. You can sputter down and you can see that the indium goes down and the iron comes up. And we've done some tests with the MRI to show where the particles go, and similarly with the SPECT CT imaging. And so we can see it out to 7 days, and that's really the key point here. And so I showed you earlier, this is the RCL01 magnetic heating material that we have got the clinical study in Spain is on. So we have a clinical study which is just about which is currently recruiting, where we're going to be injecting this into the pancreatic tumors of patients and seeking to heat those for clinical benefit. And this allows us to track those particular particles, surface radiomineralized, over 8 days here. I'm not going to go into this in detail, but this is an aspect of the work that we do. Another aspect is that we work with, there's this idea that if there's no path available to you, then you need to make your own path. And when it comes to the regulatory landscape for magnetic nanoparticles, there is a lot of unknown territory. unmapped territory. So we're working with national standards authorities around different countries in Europe to develop new ISO standards for magnetic nanoparticles. It was a big project, but one of the things that we did was we came up with terminology for magnetic nanoparticles, from single core to multi-core, and we have presented a roadmap for standardization. And one of the things that's come from that are 2 new ISO standards, one on magnetic nanosuspensions and one on magnetic microbeads. It may sound very dry, and it may sound a complete pain in the neck, and it's both of those things. But if you're in the tent, then you've got a chance of influencing, and you've got a chance of making it useful rather than horrendous. And so, for example, we have got definitions of magnetic field hypothermia and other parameters in the standard now, which we want, which we like. And we would have been very unhappy if somebody else had done it and had produced something which we didn't like. Lastly, I'm going to say a little bit about stability. This is with colleagues in Brazil and in DTU in Copenhagen. And this, I think, possibly may be of interest to the advanced materials characterization people in the audience, because what we're really interested with our iron oxide nanoparticles is whether they are magnetite or Megamites. Now, in the bulk form, they're hard to mistake. That's very obviously different. But in the nanoscale, they're really rather hard to distinguish. They are both cubic lattices of close-packed oxygen atoms, but they have a different distribution of vacancies, and they have a different distribution of charges. So the magnetite has got some ferrous iron, and the Megamites has got all ferric iron. So at the nanoscale, they're almost identical with most of the usual techniques that you use. And it matters because there's well-known redox activity associated with ferrous ions through something called the Quentin reaction. There's a highly reactive radical, the hydroxyl radical, which tends, in some circles, is thought to be a bad thing. There is also the Haber-Weiss reaction, which is a 2-step process, which can take you from Fe3+ to Fe2+ and then back to Fe3+ with the production of a radical. But as part of normal, healthy cellular function to have these reactive oxygen species in the body, they promote wound repair, but they can also cause cell death. In other words, they can have a good function or a bad function. In fact, some people are now using them as carcinogenic materials, trying to put in redox active iron oxide to actually as a cancer therapeutic. In any case, one of the things that's been missing is a good analytical method for distinguishing the magnetite and Megamites at the nanoscale. And we think that we've come up with a way to do this using Mossbauer spectroscopy. And the nice thing about it is that it's really straightforward. It's like measuring with a ruler. We look at the center of gravity of the spectrum, and we can plot that against the amount of magnetite and Megamites, and it's a very nice correlation. So I'm not going to go into this in detail, but you can use this method for even very complicated and difficult spectra. And we've done this with various types of materials. Here's an example with an anaerobic synthesis nanomaterial and an aerobic synthesis nanomaterial, and we can see over time, this is out to 500 days, 2 years, the gradual surface oxidation of the nanoparticles. So I hope I've given you a bit of a taste for what we're doing with magnetic nanoparticles, magnetic sensing, magnetic heating, and magnetic actuation, and also a little bit of a taste of what goes on behind the scenes. I've talked about the radiomineralization I've talked about our work on standards, and I've talked about our work on materials characterization. So thank you very much.
Thank you very much, Quentin, for an excellent talk. Really fascinating to see that transition from clinical, preclinical to clinical, and thank you for sticking to time. So now the floor is open. Any questions for Quentin? If you could please type in, it will be preferred. However, if you want to also raise your hand and ask a question, that's fine. I guess whilst people are gathering their thoughts, I wanted to ask you a little bit about clearance, especially in relation to endomag. What happens to the magnetic particles in situ? Have you monitored the clearance over time? And are there any kind of detrimental effects or side effects seen at all?
Well, the amount of iron that's being injected is something like 3 days worth, 3 times the recommended daily rate of iron uptake in your diet. So it's already not a very large amount. Essentially the particles are taken up by macrophages in the lymph nodes, and they're either trafficked away or metabolized in situ.
Mm-hmm. Fantastic.
So the clearance takes about a week to 10 days.
The Magna Stand project is great. It's fantastic to come up with a solid terminology, and in a way this helps to benchmark the material across labs and so on. And I wanted to know, what stage are you at with this? Is this something that's in IUPAC as well, or is this just something that's kind of used by industry and academia, or how should we look at this as it stands?
It's an interesting question about how one gets to have a standardized traceable method for measurements. We've been trying to do that with the Mossbauer method I showed at the end for characterizing magnetite Megamites. I think that you need to take it reasonably slowly, and first of all produce peer-reviewed and useful work in the literature, but that starts off by presenting the methodology, and then secondly that you have an inter-laboratory comparison that allows you to really show that it works. It isn't that straightforward. It takes a while, and then you get people on the ISO committees who are completely crazy.
Well, thank you very much for that insight. It's a really fascinating talk. Are there any questions for Quentin, please?
I've got a question.
Yep, go ahead.
Quentin. Yeah, that was fantastic. I've got a question about the your proposed COVID treatment, where you basically sort of using magnetic particles to extract the cytokines, almost in a dialysis style.
Yes.
So I mean, could you use it to actually remove viral particles, or will that not work because they don't really live in the blood, or you can't really extract enough of them? As in, what's the reason they're going after the cytokines and not the particles themselves?
We have the antibody for the cytokine. I mean, to be quite honest, that's really it's like the joke about what are you doing on your hands and knees underneath the lamplight? It was because I've lost my keys. But where did you lose your keys? Over there. But I'm looking here because I've got the light. It's what's available. I mean, ultimately it might be possible to do that, but we just don't have that right now.
Fantastic. We also have a question from James Wilton-Eli. He asks, apart from the ROS, hi James, shall I let you go ahead?
Yes, please.
Yeah, sure. Thank you. Very nice talk indeed. Yeah, just the questions in the chat. It's just about, apart from the the reactive oxygen species aspect, is there a difference between magnetite and megamite in terms of the hypothermia effect?
People say that there is. I've never really seen it. And in fact, I'm in the multi-core camp. I like the idea that you need to have the interactions between the magnetic cores in a particle to get the big heat rather than single core. But it's still quite an open area of debate.
Okay, because I'm just wondering how much design would need to go into the actual, magnetic material as opposed to the rest of the system.
There's been some really quite major changes in the last 5 years, I would say. People have started talking about nanoflowers, which are essentially exchange coupled cores. and they have roughly 5 times as much magnetic heating capacity as was previously routine. But it's a tricky business because you have to think about the whole process of injecting something into a person. You've got to get it all right. And one of the tricky things there is that the simpler it is, the better.
Right. Yeah. There's a lot to be said for simplicity, isn't there?
Exactly.
Just one last little question, if I may. Is it possible to use a magnetic field to help aid clearance, which follows on from Nazila's question?
Not that I'm aware of. No, not that I'm aware of.
Because I mean, I suppose that is presumably you've shown that you can then detect these magnetic nanomaterials, externally using a magnetic field, but perhaps being able to move them around, or at least sort of encourage them perhaps to be cleared. I don't know whether that's a possibility.
It's one of the problems with the forces is that they're bloody small.
Right. Okay.
It's really not easy.
They're fine for detection, but not for actually moving them.
I think that's one of the things that Khuloud showed, was that she put them into those 200 nanometer capsules. That was smart, because and there's a reason why diurnal beads are micron size.
Right. Great. Thank you.
Thank you very much. Are there any further questions for Quentin? If not, then I'd like to thank all the speakers from session 1. Thank you very much. We've really had an interesting, fascinating afternoon so far. And of course, we are a little bit behind time and we do need a break. So if I may suggest that we take a 10-minute break and come back at around 3 o'clock, please. That would be fantastic. And once again, thank you very much to all our speakers. Thank you.
Thank you.